mainly we will be talking about how would the information be accessed inside a gene. So primarily in this step, we call this, or the first step in this process in accessing that information would be called the transcription. So literally, for transcription, it means making a copy or creating a copy. So for the primary function of our genetic material, which is our DNA, mainly we use it for storage of information that is needed in order for us to create a certain living organism, or to create or make up, no, a living organism. So the information that is contained within units is called our genes. So quick review, at the molecular level, our gene is defined as a segment of our DNA that is used to make the functional product that is either your RNA molecule or a polypeptide. So let's go back to the question of how can we access the gene. So again, it's through transcription. So for this topic in genetics, mainly transcription would refer to the process of synthesizing your RNA from your DNA sequence. So we will talk about the synthesis of our RNA. Also, the structure of our DNA, it would not be altered as a result of transcription. Rather, the DNA-based sequence has only been accessed to make a copy in the form of your RNA. Therefore, the same DNA can still continue to store the information, and DNA replication will be discussed, no, in the succeeding chapters, wherein we will provide a mechanism to copy the information so we can transmit it from one cell to another cell, or from the parent to the offspring. So we will also encounter your protein encoding genes, which we also call as your structural genes. So we have your I'm sorry for my penmanship, but which encodes the amino acid sequences of the polypeptides. So when a protein encoding gene is transcribed, the first product would be our RNA molecule. And we would know this RNA molecule as our messenger RNA. So during the polypeptide synthesis, we will also encounter a process that we call as translation, wherein the sequence of the nucleotides within our messenger RNA would determine the sequence of the amino acids in the polypeptides. So we'll go through them one by one. So, quick question. What is the central dogma of genetics? Who knows what the central dogma of genetics is? Anybody?
Ganun lang guys, since nadumduman
Okay.
ko na sa last nga assignment. So, the central dogma says nga ang DNA hangga may process, may logical nga step uh, that leads to the final products in genetics, which is the proteins. So, halin sa nucleic acids for RNA to DNA and then to proteins. Let me check lang.
That's correct. So basically, the central dogma would be the entire process, no, from our replication to transcription to translation. So one or more polypeptides would assemble into our functional protein. So this structure and functions of proteins would determine the traits. Diba? You always need to remember that what the makeup or how it is subjected, no, or how it is moved inside, or how we arrange it, no, and how it functions would determine what the trait is. So you see here we have a figure, no, a model This model actually depicts what we call as the central dogma of genetics, or we also call this one as the central dogma of molecular biology. So, of course, we know that it was Francis Crick in the year 1958 who first enunciated, no, this dogma, and it specifically tells us about information, no, wherein it comes from your DNA to your mRNA to your polypeptide. So what you need to remember here also is that it forms the cornerstone of our understanding of genetics at the molecular level. No, that's why we refer it to as the central dogma of genetics, because it is the cornerstone, no? It is the root of understanding of genetics. So it's actually easier for you to understand molecular biology if you know your genetics. No? If you know the very root of them all, no? The very minute, the very basic detail. No? So, di na siya naghalin. What is gene? What is your genetic material? No? So on and so forth. What are RNAs, DNA? No? So what are nucleotides? So that's what we need to remember, because sila niyang aton basis, no, in learning the more complex study in may it be in genetics or biology or chemistry. So for this lecture, we will study the molecular steps in our gene expression. So the overall process by which the information within the gene is used to produce a functional product, such as, for example, we have your polypeptide. No? We will also emphasize on the transcription and modification that can occur to your RNA, no, after the transcript has been made. Now, so for, just a quick recap for our essential dogma of genetics. Now, it would talk about, number one, we have your DNA replication, wherein we make copies that are transmitted from the cell of the parent to the offspring. And then your DNA will be the one who will store the information in the units that we call as genes. Now, so your genes would contain your traits. And then we will do transcription. So we are actually discussing on the transcription part right now. We have finished replication last week. So for transcription, in layman's term, this is where we produce your RNA copy of the gene. And once we have produced the RNA copy of our gene, no, we would have your translation after that. So in creating a copy, we have our mRNA, which is a temporary copy that contains the information, and then we translate that to produce your polypeptide using the information that we have taken from our messenger RNA. No? So if you have your polypeptide will become a part of the functional protein that will contribute to the trait of the organism. It will contribute to what color of hair will you have, what color of eyes, no? The complexion of your skin, how tall would you be, no? How thin, or how, what else? Um, and all the other traits that we can see, no? Okay, so for transcription, overview here, what we need to, our objectives mainly would be to be able to describe the organization of a protein encoding gene and its messenger RNA transcript. And then we will outline the three stages of transcription. So one key concept regarding the process of transcription is the short base sequence that defines the beginning and the end of our gene. And it also plays a role in regulation of our RNA synthesis. So we will begin by examining the sequences that would determine the transcription, or where the transcription would start and where it will end. And we will also briefly consider the DNA sequences that we call as the regulatory sites that would influence whether a gene would be turned off or turned on. No? So, that is how we express the trait. No? Hindi pwedeng nga tanan, naka-on. No? There are several genes that needs also to be turned off. The second important concept uh, in the role of proteins in transcription is that your DNA sequences in themselves would just exist. No? So for genes to be actively transcribed, we need proteins to recognize that particular DNA sequences and act on them. No? So your DNA sequences cannot act on themselves. They would need their proteins to recognize them and act in a way rather that can affect the process of transcription. So we will also consider how proteins would participate later in the general steps of transcription. So to start with, we have a figure here that shows a common organization of the base sequence that we need to create a protein encoding gene. So, diba, our protein encoding gene would usually function. For example, here, we have E. coli. So for each type of base sequence, they will perform their role during a particular stage of gene expression. So the promoter would provide a site that would begin the transcription, and the terminator would specify the end of the transcription. So promoter, of course, from the term itself, it would promote or it would start the process, and the terminator, it would be the one who will end the process. No? So these two sequences would actually cause the RNA synthesis to occur within a defined location. So if you can see in our figure here, the DNA, we have our DNA, which will be transcribed into our RNA from the promoter, no, from this side to the terminator. So the proteins called transcription factors, no, we have proteins that we call as transcription factors, would recognize the base sequences in the DNA and would control the rate of the transcription. So some transcription factors would bind directly to the promoter and facilitate the transcription, while other factors would recognize the regulatory sequences or elements. No? For example, we have short DNA stretches, which involves transcription regulation. So for certain transcription factors, they would bind to the regulatory sequence to increase the rate of transcription. Whereas whereas in other cases, we have inhibition of the transcription process. So the base sequence within our mRNA are used during the translation processes. So for example, in bacteria, we have a short sequence of, or within an mRNA, which is our ribosome binding site, which would provide the location for the ribosome to bind and to begin the translation process. So they also have first codon, wherein they would start, no? Or they would also call it this is the start codon, and they would also have a stop codon, which would signal the end of their translation. Oops. So for DNA, what we need to remember here would be our regulatory sequence, which is the site to bind our regulatory proteins and to influence the rate of transcription. And the regulatory sequence, of course, it can be found in a lot or in a variety of locations. We also have the promoter site, which is the site that begins or signals the beginning of the transcript, and the terminator signals the end. No? For the mRNA, no, what's inside the mRNA, you have your ribosome binding site, the start codon, and the stop codon, and the other many codons in between. So it actually shows us, it actually shows us the common organization of the base sequence that we need to create our protein encoding gene that functions in a bacterium such as our E. coli. So, usually E. coli ang ginalantaw ta. And these two sequences would actually cause the RNA synthesis to occur within a defined location. So if you can see in our figure, the DNA is transcribed into your RNA from the promoter to the terminator. Your proteins called the transcription factors would recognize the base sequence and control the rate of transcription. No? So other transcriptions would recognize or would bind to the promoter to facilitate the start, while others will recognize the regulation to end it. So next, we have the 3 stages of transcription. So for transcription, we have initiation, elongation, and termination. So for genes, the ability of genes to produce an organism's traits relies on the molecular process of gene expression. So mainly transcription being the first step in gene expression, what happens is during transcription, the gene sequence within our DNA would be used as a template to make our complementary copy. No? So we will also talk about how the sequencing in mRNA would be translated later on after we have created our polypeptides. So normally what happens here is they would just fold and become units within our functional proteins to govern the trait. But for the 3-stage, no, namely the initiation, elongation, and the synthesis of the transcript, as well as the termination, this 3 actually involves protein DNA interactions in which proteins such as RNA polymerase, or if you can remember, our RNA polymerase, which is the enzyme that would synthesize our RNA. So they will interact with the DNA sequences. And what would cause the transcription to begin? Of course, we go to the initiation phase. So the initiation stage in the process would be our recognition step. So we also call this one as the recognition step. So the sequence of the bases within the promoter is recognized by one or more transcript factors, and the specific binding of the transcript factors to the promoter would identify the binding site for the transcription. Okay? Excuse me. So it would look like this, no? So you have your RNA polymerase, which will recognize or start the process in the recognition step, no, by binding of the transcription factor to the promoter to identify the start of the transcription process. So basically this is what happens. Now you would have our DNA, and then here comes your RNA polymerase. Now it would start the transcription process. Now, while it's undergoing the process, you can take a look. Now we are actually creating the transcript. So it's making a copy of the transcript until such time that it reaches the terminator here, wherein your RNA polymerase will release the DNA because he already has a completed RNA transcript. So basically, for the initiation, it promotes it has the promoter which would function as the recognition site. So there needs to be a promoter, because if non-existent promoter, we would not be able to start. No? Because that is the signal that we will start the initiation phase. Dila na mabind si RNA polymerase. And then the transcription factor would enable our RNA polymerase to bind to the promoter. And once it is bound to our DNA, we will denature the DNA into a bubble that is known as the open complex. So by here, you see, we call this one as the open complex because we are trying to actually untangle the DNA by we're actually trying to untangle the DNA and creating a copy, no, of the transcript. Next, we have the elongation, wherein we synthesize the RNA transcript. So your RNA polymerase would slide along your DNA in an open complex to synthesize the RNA. And then once we reach the terminator, it causes the RNA polymerase and the RNA transcript to dissociate. So as what I've mentioned, once you reach the terminator, it would automatically dissociate from the DNA. So the transcription factor and the RNA polymerase would be the first ones to bind the promoter when the DNA is in the form of your double helix. And for transcription to occur, if you can still remember, the DNA strands need to be separated. So this would allow one of the two strands to be used as a template for the synthesis of a complementary strand. And this synthesis occurs as your RNA polymerase slides along, which forms a bubble towards your terminator to create the completed transcript. So, binasulit ko lang siya, guys, in order for us to fully understand it. So, in summary, our gene is the organization or is an organization of DNA sequences. So you have your promoter would start the signal or signal the start of transcription, and the terminator will signal the end. You have your regulatory sequences that would control the rate of the transcription, and we have genes that can encode polypeptides. We also have specific start codon, stop codon, and medicodons in between in our mRNA. And for transcription, it mainly occurs in 3 steps: the initiation, the elongation, and the termination. So, muna sila. You have your initiation, you have your elongation, no? And the termination. What about transcription in bacteria? So here we would talk about the different characteristics of bacterial promoter and explain how RNA polymerase can transcribe the bacterial gene, as well as to compare and contrast the mechanisms of transcriptional termination in bacteria. So we have these two famous, or a few, not just two, a few famous people. So in 1960, we have Matthew Masselson and Francois Jacob, who found that proteins are synthesized on our ribosome. So a year after that, or let's say Jacob, no, or I don't know how to pronounce it properly, let's just say Jacob, and his colleague, Jacques Monod, proposed that a certain type of RNA acts as a genetic messenger. So from the DNA to the ribosome, something acts as a messenger to provide the information for the protein synthesis. So they hypothesized, no, that this RNA, which they called as the messenger RNA or our mRNA, is transcribed from the nucleotide sequence within our DNA and would direct the synthesis of the particular polypeptide. So the proposal was actually a remarkable insight, considering that it was made before we have actually first isolated and characterized mRNA in vitro. And in 1961, this hypothesis was confirmed by Sidney Brenner in collaboration, of course, with the original authors. Sorry, okay, so they found that when a virus infects a bacteria or a bacterial cell, a virus-specific RNA is made and rapidly associates with pre-existing ribosomes in the cell. So since they have discovered this or pioneered these studies, we have actually learned a lot more about the molecular features of bacterial gene transcription. No? So we will examine the 3 steps of gene transcription process as they also occur in bacteria. So again, let's go back to our promoter. So the promoter would be the short sequence of our DNA that is necessary to initiate the transcription. So the type of DNA sequence known as the promoter gets its name from, of course, promoting the gene expression. Or more precisely, this sequence of bases directs the exact location for the initiation of RNA transcription. And most of our promoters are actually located ahead or on the upside or upstream where the gene transcription begins. So the bases in our promoter sequence would be numbered in relation to the transcriptional site. So here in our figure, the site is the first base that is used as a template for RNA transcription and is denoted as the +1. And the bases that would precede the site would be numbered in a negative direction. No? So here, this is our transcriptional site. No? And pakadto na siya sa negative. Although our promoter may encompass a DNA sequence, by comparing them, our DNA bases with many promoters, we actually have learned that there are several sequences that are necessary for a promoter to be functional. No? So the sequence in the top DNA strand, if you can see here, we have -35 sequence. And then it's 5' thymine, thymine, guanine, adenine, cytosine, adenine, 3'. And the other one, we have here the -10 sequence, no? Which we have your thymine, adenine, thymine, adenine, adenine, thymine, no? So we sometimes also call this sequence as the Pribno box. Pribno box. It's named after its the scientist who discovered it, no? David Pribno. So he was the one who was able to discover this sequence, no? So your promoter mainly consists of from the -35 sequence and the -10 sequence containing 16 to 18 base pairs in length, and it starts from the transcriptional site, which is our +1. No? Next. So here, the conventional numbering system of promoters can be seen, and the first nucleotide, again, would act as the transcriptional site. So it's designated for +1. And the numbering to the left of this spot is in negative direction, no? So towards a negative direction, whereas the numbering to the right would be a positive. No? So for example, the nucleotide that is immediately on to the left of the +1 nucleotide should be numbered -1. And the nucleotide to the right would be +2 automatically. So when we are numbering nucleotides, we cannot find 0. There is no 0 nucleotide in the numbering system. Now, so for a lot of bacterial promoters, we can see our promoters between the -35 to -10 sequences. No? So sila sa dirita ni sila usually makitaan. Ang aton nga promoters. Are you still with me lang? Next, we have the sequences within our DNA that are found in promoters are our regulatory elements that varies among different genes. So for example, here we have a figure that can illustrate the sequences found in several different E. coli promoters. So the most commonly occurring bases within a sequence element to form a consensus sequence. So this sequence is efficiently recognized by proteins that would initiate the transcription. So for a lot of bacterial genes, the correlation at a maximal rate is still found in negative 35 to our negative 10 sequences. Okay, so our bacterial transcription is initiated when RNA polymerase holoenzyme binds at our promoter. So we have actually considered the DNA sequences that constitutes a functional promoter. Now, let's try and turn our attention to the proteins that recognizes those sequences and carry out the transcription process itself. So we have the enzyme that catalyzes the synthesis of our RNA, which we call as the RNA polymerase. In E. coli, the core enzyme is composed of 5 subunits. You have your alpha, your beta prime. I think this one should be the sigma. Let me just double check. I don't think this is correct, but I think this is, let me double check. Okay. Okay, so it's actually correct. Sigma is the sixth gali. So we have your alpha, alpha 2, beta prime, omega, and we have the sixth subunit, which is our sigma factor. So this actually creates our core enzymes, and we refer to them as our holoenzyme. So the two alpha subunits are important in the proper assembly of the holoenzyme. No? So for the alpha units, they are mostly involved, no, in the proper assembly of the holoenzyme and the binding to the DNA. Whilst our beta subunits are also needed for binding, but they also carry catalytic synthesis of our primary, or our RNA rather. We also have our omega subunit, which is important for the proper assembly. And this holoenzyme is required to initiate our transcription process. So for the primary role of our sigma factor, it is mainly to recognize our promoter. No? So take note, for our alpha units, they are for assembly and binding. For beta, they are also needed in binding, but they carry catalytic synthesis. For omega subunits, it's needed for the core enzyme assembly. And this holoenzyme altogether, no, these 6 subunits, would initiate the transcription. So the proteins such as our um, sigma factor would influence the function of our RNA polymerase. Okay, so here we have a figure of the binding of our sigma factor protein to the DNA of a promoter. So the part of the protein contains 2 alpha helices. No? So these are our alpha helices that is connected by a U-turn. No? So this is connected by a turn that is also termed as a helix turn helix motif. So we also call this as helix turn helix motif. And these two alpha helices of the protein can fit within the major groove of our DNA. And the amino acid side chains within our alpha helices would form the hydrogen bonding or the hydrogen bonds within the basis of our DNA. So after your RNA polymerase is assembled into the 6 subunits, it would bind or it would loosely bind to the DNA and would slide along our DNA. No? Just like a train. No? So you have your train rolling down the tracks. No? It's the same with your RNA polymerase going down or going through your DNA. So how do we identify the promoter again? So for the promoter part, when a whole enzyme can encounter the promoter, it would recognize, or rather specifically, the sigma factor would recognize the -35 to -10 sequence. So that's how it would know that this part of the DNA is the start, or this part promotes the start of the transcription process. So also, our sigma factor protein would contain um, our helix turn helix motif that would bind to the sequences and would fit there. No? So that's how we identify. So here, as shown in the picture that you see, the process of transcription is initiated when your sigma factor within the holoenzyme binds to the promoter. So here, sorry. Here, we have your sigma factor that binds to, or rather, your sigma factor within the holoenzyme would bind to the promoter to form the closed complex. So for the closed complex, the strands of the DNA in the promoter would not be separated, no, in comparison to the open complex. So for the transcription to happen, the double-stranded DNA must be unwounded first. No? It needs to be untangled first in order for us to create our needed open, sorry, hindi ko naman. Ay, nako. Our open complex. So here again, let's repeat it so that you won't get confused. We have your RNA polymerase holoenzyme. No? Inside can be found a sigmoid factor which will attach to the promoter region. And once you have inserted or started the coding process or the transcription process on the promoter region, it would slide along the DNA, no, to create your closed complex. So once you have created your closed complex, an open complex would then be formed right after. Because by this time, we are starting to unwind our DNA. No? So, ginabulag na sila. And the unwinding would begin at the -10 site, which would only contain both your adenine and thymine base pairs. No? So, just like here, by doing so, a short strand of RNA is made within the open complex, and then your sigma factor would be released from the core enzyme, and then the release of this factor would mark the transition to the elongation. So once your sigma factor has been released, it means that it will start to elongate. The transcript would start to elongate until such time that it reaches its terminator, wherein must stop naman siya because it would inform us that the transcription process has been completed. Gets lang? Gets lang? Dugal-lutaw na sa clouds. Wala pa man. Bala nyo lang kung galutaw na kamo sa clouds, ha? But, let's think of it this way. Anong pinaka-example man? it's very hard to create a real life example, no? Because this is very specific. So we cannot compare it to any other real life example. But just to try to expound, your understanding of how we copy and paste, inside our gene. No, this is just like basically copying, no, the genetic codes inside our body. So again, we have your RNA polymerase, and inside your RNA polymerase would be the sigma factor. Your sigma factor would bind to the promoter. So once they have bound together, they would form your closed complex. No? So sigma factor plus your promoter region, sigma factor plus your polymerase once they have reached the promoter region would form your closed complex. Mula nang tandaan nyo. Basta si RNA polymerase at si sigma factor, which we call as the holoenzyme, reaches the promoter region, which is the -35 to -10 na sequence. No? How do we identify promoter again? It should be within these numbers of nucleotides. -35 to -10 na sequence. So that's the promoter region. Once they have reached that region, they will create a closed complex. So for the transcription to happen, we cannot create or we cannot initiate transcription if the complex is closed. How can we copy if it's closed? So we have to open it. So to start the transcription, we need to unwind our DNA, diba? As what I have discussed with you with the replication part, we have to remove these two sides together. No, hindi man gid remove, but we have to untangle them. No, separate them from each other. So here, this is what's gonna happen. No, so once you have separated that, that means that you have created an opening. That's the time that we call it as the open complex. Okay? So once it has become an open complex, das ma-start na ni guys si transcription process. So for the unwinding, how can we know that the unwinding process will start, or where will it happen? Normally, the unwinding would begin with our -10 sequence site. No? Sa -10 naman nga sequence. And normally, what we can find inside that sequence would be our thymine and adenine, nga mga nucleotides. So if you can remember, Pribno, sa dili na siya ga-start. Si Pribno box, which contains only the adenine and thymine base pairs. So these two base pairs would form only 2 hydrogen bonds. That's why dili ma-unwind. Gets nyo? In comparison to cytosine and guanine forms 3 hydrogen bonds. So it would be easier to untangle your AT rich region rather than the CG region. So therefore, the DNA in your AT region is more easily separated because there are few hydrogen bonds that needs to be broken up. No? So if you ask, Miss nga, dira gid sila ma-create sang open complex because it's easier to break 2 than to break 3 hydrogen bonds. So the lesser the bonds, there is much more possibility nga pwede ka mo ma-separate, no? Just like in a relationship, no? Mas weaker ang bond, mas dasig ka mo magbulagay. Mas stronger ang bond, budlay ka mo bulagon. Okay? So the release then but if this happens, if the unwinding starts, smile-smile lang kamo da sa mga basta na sa mga luyag-luyag na, no? Interesado, you know? When the unwinding starts, we will then start with the transcription process. So this short strand of DNA that you see here, the red thingy, ribbon that you see, is now made inside the complex. And once this has started to appear, or once this starts to appear, then your sigma factor will now be released from our core enzyme. Once we have released the sigma factor, this blob right here, if you can notice, it's part of our holoenzyme, our complex. Once we have released this, kaya nag-start na transcript, i-release this automatically. It would then transition the elongation phase. Once nadulat na si stigma factor, we would know that now is the time for elongation. And the core enzyme, our RNA polymerase, may now slide along to the DNA to synthesize a strand of our RNA. Lang. Okay. Next, the RNA transcript. So for the RNA transcript, after we have initiated the transcription process, and after we have completed it, the transcript would be made during the elongation stage. So the RNA polymerase, would move along the DNA, causing it to unwind, and the DNA would be used as a template for the RNA synthesis. And then we would term it as the template strand. Sometimes we also call it as the antisense strand. The opposite DNA strand is the coding strand, which is also called as the sense strand. And it also has the same sequence as our RNA transcript being synthesized, except that the thymine in the DNA would correspond to uracil in RNA. Diba? Thymine in DNA, it corresponds to uracil in our DNA. And as it moves along the open complex, it would be formed by the action of our RNA polymerase, approximately 17 base pairs long. So in average, the rate of RNA synthesis would be about 43 nucleotides per second. And behind our open complex would be the DNA, which would rewind it back into a double helix. So in summary, the chemistry of transcription here that we see by our RNA polymerase is actually similar to the DNA synthesis via the DNA polymerase. You can still remember the previous topic that we have discussed. So your RNA polymerase would connect to the to our nucleotide in 5' 3' direction. And during this process, your RNA polymerase would catalyze the formation of your bond between your 5-phosphate group on one side of your nucleotide to your 3' hydroxyl group on the previous nucleotide. So here, this figure actually shows us the different names of each part. So our coding strand would start here sa aton nga 5', and this would be our template strand. Your template strand is where makopy kita sa aton nga RNA, sa aton nga gina-transcript. So this is actually what happens in the in specific or in details. So if you want to enlarge, the template strand would look like this. So you have your coding strand, the blue. You have the template strand. The template strand is what we use to copy. So your RNA would actually have similar nucleotides with the coding strand. The only difference is that instead of thymine, you have uracil because it's RNA. It's not DNA. Okay? So every time na ma-finish na ka create sang template creates ang imong RNA from your template strand, it would be rewinded back, no, into the helix. There is rewinding. See here, it would reattach itself into its coding strand. And you also have here, on the other side, is the unwinding. This is where you remove the hydrogen bonds. And the direction of the transcription would be towards 3'. So it's 5', 3'. And here is where your nucleotides will be added to the to the RNA strand. No? So diri na sila gina-attach. So diri man nagasulod si nucleotide or the nucleoside triphosphates. So here we have another example in the case of transcription of multiple genes within a chromosome. So the direction of the transcription and the DNA strand used as a template would vary. But here in this figure, it shows 3 genes adjacent to each other within a chromosome. Your genes A and B, this one and this one, are transcribed from left to right, and the bottom DNA strand is used as your template. By comparison, we have here gene C, which is transcribed from right to left, and the top DNA strand is used as a template. So if we try and take note in all 3 cases, the template strand is read in 3', 5' direction, and the synthesis of the RNA occurs in 5', 3' direction. What about transcript termination? For transcript termination, it's done either by an RNA binding protein or an intrinsic terminator. Of course, in the natural order of things in the world, there needs to be something that says stop. Na? There needs to be something that would inform you to stop if kinanglan na mag-stop. If it's something that tells you to start, no, we also have that. So if we actually try and take notice of the very minute, tiny details, no, that we can see in the universe, we follow the same thing, no. We follow the same rule, the same law. We have the time where we start and the time where we stop. So for the termination part, the end of RNA synthesis is also referred to as termination. But prior to the termination, the hydrogen bonding between our DNA and RNA within the open complex is of central importance, because we need to prevent the dissociation of our RNA polymerase from the template strand. So the termination would occur when this short RNA-DNA hybrid region is forced now to separate. Thereby, we will release the newly made RNA transcript. So, for example, in our E. coli, we have 2 different mechanisms for terminations that we have identified. Now, so, for the types, our RNA binding protein or an intrinsic terminator. So, we have your para-dependent termination. So, in para-dependent termination, it requires 2 sequences, while for the independent, the process of independent does not require your paraprotein. So in this case, the terminator is composed of 2 adjacent nucleotide sequences that would function within our RNA. So here is how it happens. So for the dependent termination, it would require 2 sequences. So first, a sequence upstream from the terminator that we call as the root site. This one. I'm sorry. This is our root site. So the root site is for row utilization site. No. RUT meaning row utilization site. And it acts as a recognition site for the binding of our row protein. So how does the paraprotein facilitates the termination? So what happens here is it would act as your helicase which is an enzyme that can separate your RNA from the DNA hybrid region. Now, so it's like a baby, or it's like a son or a daughter, or it's like you. No, eventually you will be removed from your parents. No, you will move on from your mother, from your father, from your family, and be independent in the world. So that's what also happens with RNA once it has completed its transcript. So later on, if mag-graduate na kamo, makapasar na kamo sa board, you would start to live independently like our RNA transcript. So your helicase again would separate RNA DNA hybrid region after our RUT site is synthesized in the RNA. You then would have your paraproteins that would bind to the RUT site in your RNA and would move towards the 3' end. No? So the second component of our para-dependent termination is the site where the termination actually would take place. So this is our terminator or the termination site or the terminator site. So the DNA would encode your sequence that can contain your GC base pairs that forms your stem loop. So this stem loop would almost immediately after your RNA sequence is synthesized and quickly bind to RNA polymerase would result in a conformational change that would cause RNA polymerase to pause its RNA synthesis. So the stem loop would actually signal the stop of synthesizing. So the pause would allow your paraprotein to catch up to the stem loop and pass through it and break the hydrogen bonds between your DNA and RNA in your open complex. So when this occurs, the complex RNA strand would then be separated from the DNA along its RNA polymerase. So here, so many ang step guys. 1, 2, 3, and 4. No? So you have your para recognition site, which is the rod. No? So your rod would bind, or rather your paraprotein would bind to the rod site to move and moves towards the 3' end. No? So you have your paraprotein that would transcribe it and create your stem loop and then proceed to the terminator. Once you have created the stem loop, it would signal a pause. Once nga nag-pause na, it would signal the separation. Okay? So, amala na siya. Individually, nakaseparate na sa each other. You have your DNA, your transcript, your holoenzyme, and your paraprotein. What about independent termination? For independent termination, it does not require your paraprotein. So in this case, the terminator is composed of 2 adjacent nucleotide sequences that can be found in your RNA. One, you have your U-rich or the uracil-rich sequence that can be found in the 3' end of our RNA. And the second sequence is adjacent to our U-rich sequence and would also promote the formation of a stem loop. There is also a stem loop formation. So you have your NUS-A and the U-rich. So the pausing would be stabilized by other proteins that bind to RNA polymerase. So for example, a protein called the NUS-A, which will bind to our RNA protein, or RNA polymerase rather, to promote the pausing at the stem loops. And at the precise time, the RNA polymerase would also pause the uracil-rich sequence that is found in our DNA strand. So basically, the difference is that it doesn't have your paraprotein. So pag untangle na sila, you would have all this. So reminders, no? Or what we need to take away Many bacterial promoters have sequence elements at -35 to -10 sites, and the transcriptional start site would always be at +1. And during the initiation phase of transcription in E. coli, we have your sigma factor, which is bound to our polymerase that can bind to the groove of the DNA to recognize our sequence elements. And this would form our complex process, wherein we would follow to have our open complex and would release our sigma factor to proceed with the elongation phase, no? Until such time that we reach our termination phase as well. Okay. Let's take a quick 5-minute break. You can go to the bathroom or drink water for 5 minutes. Hello, guys. Is everyone back? Okay, let's continue. Are you back? Okay, tutay na lang gada. A few more slides. Okay, so let's continue with transcription in eukaryotes. So we will talk about the functions of the types of RNA polymerases in the eukaryotes, the characteristics of a eukaryote promoter for a protein encoding gene, and the open complex. So for this objective, um, we will also deal with the many basic features of gene transcription that are very similar in bacterial and eukaryotic species. So much of our understanding of transcription, of course, starts usually with the study of yeast and other eukaryotic species, including mammals as well. So in general, gene transcription in eukaryotes is much more complex than it is in bacteria, and our eukaryotic cells are larger than our bacterial cells and contain a variety of compartments that we know as organelles. So this added level of cellular complexity would require that eukaryotes make many more proteins and consequently would have many protein encoding genes. So also for most of our eukaryotic species, they would be multicellular, composed of different cell types. And because of this multicellularity, it adds a requirement that the genes need to be transcribed in a correct type of cell and during the proper stage of development. So we have the first one here, which is the multiple RNA polymerases. So for eukaryotes, we have multiple RNA polymerases that are actually structurally similar to bacterial enzyme. So the same structure, it's just that they have, we have multiple numbers. So the general material within the nucleus of eukaryotic cell is transcribed by three different RNA polymerase enzyme. So you have the designated RNA polymerase 1, your RNA polymerase 2, and the 3. So what are the roles of these enzymes? Each of the three RNA polymerases would transcribe different categories of genes. For the one, it would transcribe the gene for ribosomal RNAs, except for 5sRNA. For our 2, it would transcribe all protein encoding genes, and it would be the one who will be responsible for synthesis of our messenger RNAs. It would also transcribe the the genes rather, for most of our snRNAs, which are needed for RNA splicing, which we'll discuss in the next few chapters. So in addition to this, it transcribes several types of genes that produce other non-coding RNAs and includes most long non-coding microRNAs and snRNAs. While for the polymerase 3, it would transcribe genes for tRNAs and genes for 5sRNA. So here is the comparison between your bacterial RNA polymerase and a eukaryotic RNA polymerase that can be found in a yeast. So all three of our polymerases are very similar to each other, and they are also composed of many subunits. So mostly or mainly they contain two large catalytic subunits similar to the beta and the beta prime subunits of the bacterial RNA polymerase. The structures of RNA polymerase from a few different species have been determined by X-ray crystallography for this case. So if you notice, the similarity is very remarkable. No? And this structure also provides us a way to envision how transcription works. Diba? So if you notice, it really looks like a hand, no? Something like this. So for the crystal structures, no, we have your RNA polymerase on the left and the eukaryotic one on the right. So the bacterial enzyme, this one was taken from Thermus aquaticus, while the other one was taken from Saccharomyces cerevisiae. So as seen in the figure above, we have DNA, which enters the enzyme through the jaw lines, through the jaw, and lies on the surface within the RNA polymerase that we term as the bridge. So the part of the enzyme that we call as the clamp is the one who controls the movement of the DNA through the polymerase. And then a wall in the enzyme would force the RNA-DNA hybrid to make the right angle turn and would bend uh, would the bending rather would facilitate the ability of the nucleotide to bind to the template strand. So basically the process is just the same here. So your wall in the enzyme forces, or rather the magnesium that can be found in the catalytic site, and your NTPs will also enter your catalytic site via the pore. So this is our pore. So the correct nucleotide would bind to the template DNA, just like the same with the bacterial one, and would attach to the 3' end. As your RNA polymerase would slide along your template strand, a rudder, which is about 9 base pairs away from your 3', would force your RNA-DNA hybrid part, the single strand apart, and the single-stranded RNA would exit under your small lid. So the structure for both, actually, they really look the same. It's just that in this diagram, the direction of the transcription is from left to right. So the double-stranded DNA would enter the polymerase along the bridged surface that is between the jaw and the clamp, and at a region termed the wall, it would bend into a right angle to enable the nucleotide to bind. So if you can see, it basically looks the same. But the terms are different lang. We have the bridge, the clamp, and the rudder. Your catalytic site, which has your magnesium. And this is where the NTP would enter, so that your nucleotides can attach to our RNA transcript. We also have a core promoter and regulatory elements for eukaryote. So for the eukaryotic protein encoding genes, there are at least 3 features found in most promoters. We have your regulatory elements, we have your Tata box, and the transcriptional site. So here in the figure, it actually shows a common pattern of sequences that are found within the promoters of the eukaryotic protein encoding genes. So in the picture, we can see the transcriptional start site, this one. This is our transcriptional start site, and the site usually occurs at adenine. And then 2 pyrimidines, or it can be either a cytosine or a thymine. And a cytosine would precede the adenine. You would also have 5 pyrimidines that would follow it. And then your Tata box, which is approximately 25 base pairs in length, would be found upstream from the start site. However, the sequences that constitute eukaryotic promoters are quite diverse. So the regulatory elements that which will be discussed more later would also vary in the locations, but are often would be found between -50 to -100. So a single upstream regulatory element is involved in the binding of our RNA polymerase 1 to its promoter, whereas 2 regulatory elements called A and B boxes would facilitate the binding of your RNA polymerase 3. So the core promoter is a relatively short DNA sequence that is necessary for transcription to take place. And it consists of a sequence, this one, the TATAAA sequence, which we also call as the Tata box, and the transcriptional start site where the transcription begins. So all of this would be termed as your core promoter. If it is missing from the core promoter, the transcription start site becomes unidentified, and your transcription can start at a variety of different other locations. So the core promoter here would be produced at a low level of transcription, and we will term it as the basal transcription. So for the regulatory elements of our eukaryotic, um, we have DNA sequences that are short and would affect the ability of your RNA polymerase to recognize the promoter to begin the process of transcripting. So the proteins here that can influence the rate of transcription would be the transcription factors, and we have two regulatory elements. So we have your enhancers, the one that we need to stimulate the transcription. And in the absence of our enhancers, most of our eukaryotic genes would have very low basal transcriptions. We also have in certain conditions that we need to prevent transcription. So we term this as the silencers. No? So for silencers, they are the sequences that would inhibit the transcription. So for the DNA sequence, such as your Tata box, for example, your enhancers and your silencers will exert their effects only at a particular gene. So they're called as the cis-acting elements, and the term cis, if you can still remember from its nomenclature, when we talk about cis, we mean next to. No? So for our cis-acting elements, though possibly they can also be found away from our core promoter, but they are always found within the same chromosome. So as the gene would be regulated, your transcription factors would bind to these elements that are also called trans. No? Trans meaning across. The transcription factors that control the expression of the gene are encoded as well. Now let's proceed to the initiation. For the initiation part, now the transcription of eukaryotic protein encoding gene, it would be initiated when your RNA polymerase 2 and general transcription factors binds to the promoter sequence. So we have also considered the DNA sequences that would play a role in the promoter of eukaryotic protein encoding genes. So we have here the 5 different proteins that we call as the GTFs, that are always needed for RNA polymerase 2 to initiate the transcription of our protein encoding genes. So it describes the assembly of your GTF and your RNA polymerase 2 at the Tata box. The Tata box is the location. So here we have a series of interaction that would lead to the formation of the open complex. So basically the process is the same. Mas complex lang sa eukaryote kay mas damo siya agyan. No? So you have these general transcription factors. First, the TF2D. The transcription factor 2D first would bind to your Tata box to play a role in recognition and promoting the core. Once you have done that, for the TF2D, by the way, it is also composed of several subunits. So the subunits would also include the Tata box, the TBP, the Tata binding protein, your RNA polymerase 2, and several other proteins that we call as the TBP associated factors, or the TB or the TAFs. Sorry. After your TF2D would bind to your Tata box, it will associate with the TF2D and would promote the binding of the RNA polymerase to the TF2F promoter. Once you have your TF2E and TF2H bind to the complex, the binding will then complete the assembly of the protein to form our complex, which is the closed one, which we termed as the pre-initiation complex. So your TF2H would now play a major role in the formation of the open complex, and several subunits would act as a helicase that can break the hydrogen bonds, because we need to understand that we need to break the bonds before we can initiate the transcript itself. So between the two strands of the DNA and thereby promote the formation of the open complex, we will also hydrolyze another subunit and phosphorylate a domain in RNA polymerase 2 that we know as the carboxyl termination domain. So therefore, your RNA polymerase 2 is now free to proceed to the elongation stage of the transcript. So if you try and compare, the same lang din sila yung gyapon. But different lang nga factors ang in play or at play. So here, try and look at it. This is how it happens for the eukaryotic. No? So you have your TF2D, which would bind to your Tata box. So your TF2D is a complex of protein that includes Tata binding protein, several other factors, no? And once it has bound to the TF2D, it would promote the binding of the polymerase to the core promoter. So once we have our RNA polymerase 2, your TF2F would now also bind to the RNA polymerase 2. So this entire complex is called as the pre-initiation complex. So it's composed of the TF2D, TF2B, TF2F, TF2H, and TF2E, together with your RNA polymerase. And your TF2H would act as your helicase to form as the open complex, and it would also phosphorylate your CTD and RNA polymerase 2. So once it has done that, the phosphorylation would break contact between your TF2B. So it will disintegrate your TF2B, TF2E, and TF2H, which will signal their release. Once they have been released, we will start to form the open complex, and the transcription process would now start. So these are actually the steps that leads to the formation of the open complex. Now, once the strands of the DNA are separated at the Tata box to form an open complex, the open complex would now move to the transcriptional start site, which is about 25 base pairs away from our Tata box. We also have what we call as the mediator. So in addition to our GTFs, our general transcription factors, we also have the mediators. And together with your RNA polymerase 2, a mediator is actually a large protein complex. This was discovered first by Roger Kornberg and his colleagues in the year 1990. So they were also awarded the Nobel Prize in Chemistry for the study of molecular basis of eukaryotic transcription. So the mediator derives its name from the observation that what it does is to mediate. So it mediates. And it mediates the interaction between our RNA polymerase 2 and our regulatory transcription factors that can bind or enhance or silence. So it would serve as an interface between our RNA polymerase 2 and many other diverse regulatory signals. So the subunit composition of mediator is very complex, and therefore it would play a very pivotal role in the switch between transcriptional initiation and elongation. Lastly, for the eukaryotic, we have the termination part. So for the transcriptional termination of RNA polymerase 2, it occurs after your 3' end of the transcript would be cleaved at the polyadenylation signal sequence. So here, this is our polyadenylation signal sequence. So as we have discussed before, your RNA in the eukaryotes are modified by the cleavage near their 3' and the subsequent attachment of the string of adenine nucleotides. So the process, which is called polyadenylation, requires polyadenylation signal sequence that directs the cleavage of the pre-mRNA. So here, this is where our polyadenylation starts. So this is actually a simplified version. So we'll just show you the photo. So your RNA would now be cleaved just past your polyadenylation signal sequence and would continue to transcribe the DNA. So we also have two models that have been proposed for transcription of termination when they were just studying this. So we have one which we know or we call as the allosteric model, and we have another one as the torpedo model. So it's actually trying to tell us, no, if how termination happens. For the allosteric model, the RNA the RNA polymerase would become destabilized after it has transcribed the polyadenylation signal sequence, and eventually would just dissociate from the DNA. So the destabilization may be caused by the loss of proteins that functions as elongation factors or by binding of proteins for the termination factors. No? So it's not shown here, but they're just saying that there is just simply dissociation because of the release of the elongation factors. We have the other model, which we call as the torpedo model, which suggests that RNA polymerase II is physically removed from the DNA. So according to this model also, the region of the RNA that is downstream from the polyadenylation signal sequence is cleaved by an entity that is called an exonuclease. So it's saying that the exonuclease would degrade the transcript in a 5' 3' direction. When the exonuclease catches up to the RNA polymerase, it would cause RNA2 to dissociate from the DNA. So this other theory informs us that it actually puts a stop because it dissociates the RNA polymerase II from the DNA entirely. Which of these two models do you think is the correct one? No? What do you think? Which would be the correct type of termination for eukaryotes? The allosteric model, wherein it just destabilizes and dissociates, or the torpedo model, wherein there is an enzyme that catches the RNA polymerase II and causes it to terminate? Actually, until now, this has not been answered, no? So we still need to study it more and more and more. So the researchers and scientists are still battling over this. Evidences that points that these two models are not mutually exclusive. So both mechanisms can actually play a role in the termination process. No? So there is no specific transcriptional termination for eukaryote. No? So either mechanism can play a role in termination. So we don't have a definite answer even until now. No? Basta mag-scientist kamo pa maka-discover sina no? How the termination or what is the specific process in terminating eukaryotic transcription. So in summary, eukaryotes use RNA polymerases 1, 2, and 3 to transcribe different categories of genes. For prokaryotic and eukaryotic RNA polymerases, both of them would have similar structures. Eukaryotic promoters have a core promoter and a regulatory element such as your enhancers or silencers. And for transcription of protein encoding genes in eukaryotes, it would require your RNA polymerase, the general transcription factors in your mediator. The 5 general transcription factors in your RNA, when assembled together, will form your open complex, and the termination may proceed according to either allosteric or torpedo model. Okay, so do you have any more questions?